All right, so this is evidence for evolution part 2. I kind of teased this at the beginning. I said we would get back to evidence for evolution um, because we need to talk about a couple more details on some of the evidence we mentioned when we were first going over like the different categories of evidence. So number one, fossil evidence. You can have a couple different kinds of fossil evidence. So you can have petrified or preserved specimens inside things. So in ice, in amber, in sand, or in clay. Here's an example in amber. Some of the fossil evidence that we have are going to be from extinct organisms. Some are going to be from not extinct or extant. So if it's something that is still alive, we would say extant organisms. But it's going to be the remains of hard parts for the most part. If it's in something like amber, we can sometimes get the remains of soft tissues. But if it's in things, if it's a fossil in rock, which is what we normally think of, it's going to be hard parts. So it's going to be bones, or sometimes it can be traces. So sometimes you'll see like impressions. It looks like there might be an impression fossil here, but most of the times it's going to be these hard parts. So bones, plates, any part of the organism that would have been hard. Because of this, we're going to see very little fossil evidence in like the plant world. A lot of that will be like impressions, and because those are harder to form, we won't see quite as many of those kinds of fossils. All right, and just a reminder between relative dating and radiometric dating. So relative dating is just like what layer of rock is it in. But radiometric dating, this is also called carbon dating because we use carbon when we're looking at living things. And it looks at the amount of C14 compared to the amount of C12 in a fossil, and it can use that to date it because over time, the C14 will break down into C12. And this takes, thousands of years. And based on the ratios that they would expect, they can tell how long this thing has been dead versus when it was alive because of that decay. Living things will not have this decay happen because they're continuously incorporating the C14 that's in the atmosphere into their bodies. Now, one example of fossils combined with this like dating is horse evolution. So here we have our fossils of the 4-foot, and we can see that the earliest horse, the earliest thing that we would consider, Hyracotherium, dawn horse, had like 5 of these bones. So we have 2, 3, 4, 5 in the 4-foot. So we've already mostly lost that first one. We may have like partial remnants of bones up here. But then as we go up, Myohippus, we have 2, 3, and 4 that are still elongated down to the bottom. Continuing 2 and 4 are getting smaller because they're not used. 3 is getting more robust. And then we have more modern horses. So this is Przewalski's horse, and then the modern thoroughbred would have the same basic bone structure. So we can look at these bones, we can date them using this carbon-14 dating, and we can see the age of these different fossils. All right. And I should mention, not all fossils, not all things are going to be able to be carbon dated. It's going to depend on a couple of different things. We're not going to go through and do like details on the carbon dating in this class. All right, genetics. This is our other evidence. Remember the gene pool, that whole thing we just did with Hardy-Weinberg, looking at our gene pool. It's the entire collection of genes in a population, and we could actually look at genes and we can see how things have changed over time. This part of the evidence is what we looked at in our biochemical evolution lab. So this was where we were looking at comparing the hemoglobin protein, so comparing actual amino acid sequences in different species. We could also do this with DNA sequences because remember, we go from DNA to RNA to protein, and so we can use that as well. Next is comparative biochemistry, and since we haven't done a lab on this, I'm going to talk a little bit more about this. But this is where we look for similarities and differences in specific macromolecules. And so some examples, antibodies or the complement system, these are both part of the immune system. Because they're part of immunity, they're both going to be pretty important. We can look at hemoglobin if we're going to track things that have like a circulatory system, and we can look at how hemoglobin compares amongst different organisms. And then actin. Actin is one of these things that's in all cells. It's part of the cytoskeleton. And so if we compare actin amongst organisms, we can look at like a lot of variation because we can look at things that are single-celled versus multi-celled. Now, depending on how much the thing is used, so how important is it that the protein structure remains the same in order for the thing to stay alive? We may see fewer differences if it's very important for it to be conserved or the same structure. So for that reason, we may see more differences in antibodies because having variation in antibodies is sometimes a good thing for immunity, but not very much like variation in actin, because in order to have a cell with a cytoskeleton, you have to have actin that is functional. Now we are going to talk about anatomy. There's a couple different ways we can look at anatomy and look at evidence for evolution. So first, there's similarities in characteristics resulting from common ancestry. So here we have forelimbs, all things that share common ancestry. They have similar structure, similar embryological development. So in the embryo, they look about the same, but they can have different functions. So we have similar bone structure. You can see the bones are highlighted. Same number of bones, similar bone structure, where we've got the one bone in the like upper limb and the two bones in the lower portion of the limb, and then we have the what would be the finger and the thumb bones down here. So similar bone structure, very different functions, right? We have the bat's wing for flying. The whale is going to use that flipper for swimming. The cat is going to walk in a very different way than the human is going to use its hand. So evidence of a common ancestor, but branches off from that four-limbed common ancestor, from that tetrapod, into lots of variation that gives us these different things. We can also look at analogous structure. Now, analogous structure is separate evolution of similar structures. So they have similar functions, similar external form, but different internal structures and development. That indicates a different origin of that same kind of concept. So no evolutionary relationship. So don't be fooled by the similar appearance. This is basically solving a similar problem with a similar solution, but in a different way. For example, our dragonfly wings and our bird wings. You could say the same thing with bat wings. Bat wings are going to be a little more similar to bird wings because they both would have come from that tetrapod ancestor. But the way that these wings are going to be managed are going to be different, right? The bat wing is going to be like skin, whereas we have feathers in the bird in order to like have that same function. So they don't have exactly the same evolutionary relationship, but they both have the same, they both are going to have bones because they come from that common ancestor. So batwing and birdwing, some evolutionary relationship because they have those same bones. Now look at our dragonfly wing here. There are no bones in there. It does not come from that common ancestor. So we have very, very different structure, but it still does that, or very different internal structure, but it still has that same like external shape that allows for the same external function for flight. All right, so here we have an example of homology, so all the bones in the different limbs, versus analogy. So this is a fly's wing. So fly's wing with bird wing, analogous structures, not homologous structures. Another thing we can talk about with anatomy is vestigial structures. Vestigial structures would be things like the appendix. Right now, you don't really use your appendix. It only becomes a problem if it gets inflamed, and then an inflamed appendix is basically an infection, and it can burst and cause all sorts of other issues, and usually it's removed in that case. The surgery does have risks, like all surgeries, so we don't just remove appendixes without them having issues, but they're not really a necessity. Another example would be our ostrich wings. Ostriches are not flying around anymore, but they have those wings because of that past. All right, some more evidence. Mimicry. These are structural adaptations that enable one species to resemble another. So a species that looks like another species in order to like distract a predator, so that it doesn't become prey, something like that. Camouflage. which is adaptation that allows a species to blend into the surroundings. And then also antibiotic resistance, which we talked about this at the beginning, so I just wanted to remind you, this is another example of that natural selection that we can see that's more evidence for evolution. This is just another, like I talked about antibiotic resistance at the very beginning. I'll kind of go over it in general again because it's a really great, easy to understand example. And use a different picture this time because that's always good for our brains to see things in different ways. All right, so here we have a bunch of bacteria, including one that just happens to be resistant. It just happens to be resistant because it has a mutation, okay? It's just random. They all get bathed in antibiotics. Most of the normal bacteria die. The resistant bacteria then, though, can multiply and they can become more common. Eventually, the entire infection could evolve into that resistant strain, and then you would need to treat with a different antibiotic in order to kill those resistant bacteria. All right, here's a picture of mimicry. So I chose these pictures because it's easier for us to see like that this is a butterfly here. But the butterfly has these spots that look like owl eyes because the things that eat butterflies are eaten by owls. And so if it can pretend to be an owl, then maybe it won't get eaten by other things because the things that might normally eat that butterfly are going to run away. Here's an example of a couple examples of camouflage. So here we have our amphibian. Lots of amphibians have different kinds of camouflage to blend in because they make good snacks for birds. So they have to kind of blend in order to not become prey. So this one is blending into the tree. And then here we have some little seahorses that are blending into the coral and the other they can blend in. There's different species that can blend in with other aquatic like plants and algae and things. So those are really cute little guys. So for practice, and this is your assignment for today, find an example of either a vestigial structure, mimicry, or camouflage and turn in a web link where it is described or pictured so that I can see the really cool example that you found. All right, that is it for this lesson. Enjoy.